Okay, hello everybody. Thank you so much to IADA for the chance of presenting our research today. So I'm a second-year PhD student at the Tokyo University of the Arts, and I will be presenting today on the topic of identification of sepia inks for the use of DNA analysis. So ink identification, as you all will be full aware of, is a very difficult topic. And while there has been a lot of progress on the identification of metal chelate inks like logwood and iron gall, the identification of purely, is it too loud? I'm so sorry. Okay. The identification of purely organic inks that do not contain any significant amounts of metal ions, such as sepia, still remains very challenging. And we hope that for this, we might be able to contribute to their identification by looking at the DNA contained in the sepia inks. So for this presentation, first I want to start with a quick introduction of the other people involved in this project before getting into the topic itself. First I will be giving a quick introduction into sepia as a material before moving on to the methodology we used for the identification in this study. There I will be explaining some of the basic principles the analysis method is based on before moving on to the results that we were able to obtain so far. I will then be ending on a quick summary as well as giving a short outlook into the next few projects into the next few steps of this project that we are planning. So this research is part of my ongoing PhD thesis at the Tokyo University of the Arts under the guidance of Professor Tsukara, who is the head of the Conservation Science Laboratory at the Tokyo University of the Arts. Additionally, I have also been receiving generous support and guidance from Professor Oshikane, who is an expert in protein chemistry and DNA analysis and has been providing the infrastructure and the expertise required for performing the analysis. So with this, I want to get into the main topic of my presentation today, sepia ink and its identification through DNA analysis. So sepia is a fine art material made from the black secretion that is naturally produced by squids and other cephalopods. The existence of this secretion has been known at least since antiquity. However, it does not seem to have been used as an art material until the later half of the 18th century. So to make the ink, you take the dried ink sacs of the animals and you grind them into a fine powder and add a strong alkali and boil the suspension. You filter it and then you add a strong acid to the filtrate, which causes the contained eumelanin to fall out of the dispersion and settle on the ground as a precipitate. And then you can decant this precipitate, wash it and mix it with binder to be used as an ink. As a powder, the sepia appears almost black, very similar to carbon black. However, when it's applied to paper, it has a brown hue that may vary slightly depending on the source material and the preparation method. So chemically speaking, squid ink mainly contains eumelanin as its main colorant, and eumelanin is a complex biomacromolecule consisting of two monomers, dihydroxyindole and dihydroxyindole carboxylic acid. And one of the most important properties of eumelanin is the broadband absorption it exhibits over the entire UV and vis wavelength spectrum, as well as its ability to transfer that photon energy into heat at a very fast rate. So this broadband absorption and heat transfer makes both Raman and FTIR analysis very challenging. And even if you are able to get a spectrum, oftentimes it does not exhibit many characteristic peaks, and the analysis becomes even more difficult when the ink is applied to paper. So there have been some successful approaches of identifying sepia using Raman and GCMS. However, it still remains a very challenging material to identify, and here we hope that through the use of DNA analysis, we might be able to contribute to its identification. So with this, I want to get into the main methodological part of this presentation, the PCR and DNA barcoding and its application for sepia inks. So while there have been other cases of the application of DNA analysis in the book and paper conservation field, it is still a rather uncommon method for the field. And so I would like to take a bit of time to summarize the principles that the method is based on before getting into the results that we've been able to obtain so far. So DNA is the biomacromolecule that carries the genetic information for all living organisms, and it contains 4 different types of bases that form characteristic base pairs, and the sequence and order of these base pairs are specific for each species. So by focusing on characteristic sequences, we can identify the species in question. So there are several types of DNA analysis, and many of them require highly specialized laboratories with ultra-clean rooms and very sophisticated equipment. However, the method that we have been using in our study can be done even if there are contaminant DNAs contained in a sample, and therefore can be done in most laboratories using only these 3 pieces of specialized equipment. So in principle, it uses the same method as the PCR tests that were used during the 2020 COVID pandemic to track infections. So we take a sample that may or may not contain the DNA that we're looking for, and then we define a specific region within that DNA, and we use PCR or polymerase chain reaction to make copies of that DNA sequence. And then through DNA barcoding, we can separate the samples based on their molecular weight and visualize all DNA contained in the sample using a fluorescent dye and UV light. So how does the PCR work, and why is it so specific for certain species? So PCR is a chemical reaction that allows us to copy only very targeted sequences of DNA. And this is possible through the use of so-called primers. So primers are short synthetic pieces of DNA that are designed to fit into a very specific region within the target species DNA. And so in our case, we're focusing on squid DNA. And practically speaking, what that means is we place a sample which might contain the DNA that we're trying to identify into a reaction mixture. And inside this mixture, everything is contained for the reaction in order to take place, including 2 types of species-specific primers. We then place the solution into this type of thermocycler, which is basically a small oven which heats and cools the samples to very specific temperatures for specific intervals. So during the heating and cooling cycles, the reaction summarized in this slide, in this illustration, is taking place. So first, during the heating, the DNA that is contained in the sample is denaturized and split into 2 individual strands. At this point, our target-specific primers attach into the target regions, framing our region of interest. Then, as the solution cools down further, an enzyme that's contained in the solution helps to extend the molecule starting from those primers, giving us 2 double-stranded copies of our 1 strand that we started out to begin with. And by repeating this cycle over and over again, you can have exponential growth and exponential increase of your target DNA within your sample. So the reason this method is so specific is because the primers are designed to specifically only be able to attach to certain regions within the DNA. Even if there are small mismatches, it will not be able to bind. And so any other DNA that's not our target will only be denaturized during this step and not amplified. After amplification, all the DNA that is contained within our sample can be separated according to its molecular weight. Because the primers that we chose for our solution let us specifically target certain regions, we know exactly how long those regions in our DNA should be. And because only that region will be amplified, it is present in much, much higher concentrations than any other DNA who has only been denaturized. So the amplified DNA samples are then loaded into an agarose gel and are separated using electrophoresis. Since the DNA is slightly negatively charged, it will move through the gel towards the positive cathode, and the movement of the speed is dependent on the size of the molecule, which allows us to separate it. After the separation has taken place, we add a fluorescent dye to the gel, and we can then visualize the separated samples using a UV camera and UV light. And because we know exactly how long our target species should be, we know exactly where to look for it in our sample. And so if there is a more intense band at that specific length marker, we know that our DNA is contained in our sample. There are a lot of factors, however, that can adversely influence PCR reactions, such as the degradation of the DNA. Like if the DNA has degraded to a point where the snippets are too short, we might no longer be able to amplify it. And the presence of PCR inhibitors can also adversely affect the reaction from happening. And PCR inhibitors could be a lot of things that might also be contained in artworks, such as salts or metals. So to determine whether this method is a valid approach for the identification of sepia inks, we performed some experiments on a set of mock-up samples. For the sample creation, we used 2 different types of ink, commercially available crema sepia, as well as an ink that we prepared ourselves in the lab using frozen squid ink according to a historic recipe. As a substrate for our samples, we used Whatman filter paper, and the inks were applied with a brush, and half of the samples were artificially aged using thermal aging. The swabbing, or the sampling, was conducted using commercially available cotton swabs. To give some indication of how much pressure you need to apply during the sampling, we did the sampling on a set of scales, just to give some number behind the pressure that we applied. And after sampling, all the swabs were recorded using a microscope, so we could later compare the successful and unsuccessful PCR results with the amount of sepia, visible sepia that is stuck to the swab. So basically we then take the tips off of the cotton swabs and we place them into sterile tubes, and we add distilled water to the swabs to extract the DNA from the swab. They're then centrifuged and vortexed, and 1 microliter of these solutions then becomes the sample DNA for our PCR. We then add the sample DNA into the PCR master mixture containing our squid-specific primers and incubate the samples in the turbocycler. So after the PCR, we separated and visualized the bands as I described before. And so for clarification, if there is no DNA present, or no target DNA present, we will have either no bands or like very unspecific blurry bands at random intervals from DNA that has just been degraded. However, if the DNA is present in our sample, we will have these clear bands at the target length marker. So with this, I want to get into the results that we were able to obtain so far. After positive initial results that we got from liquid inks and from performing different sampling experiments, trying out different types of swabs. We performed the analysis on our aged and non-aged samples using the cotton swab sampling method, the results of which you can see here. So these dark boxes here at the 200 base pair marker are indicative of the presence of squid DNA within our sample. So if we look at the results one by one, we can see that the unaged crema sepia yielded the most easily and clearly visible bands, allowing for an easy identification of all samples. The sepia that we ourselves produced in the lab also has bands at all also showed bands for all samples. However, they were much lighter and less distinct, indicating that the DNA contained is either more fragmented or some other PCR inhibiting component is contained in the sample. The artificial aging generally led to a weakening of the bands. So in the case of the crema sepia, this means that we were still able to identify all the samples as containing squid ink. However, some of the bands have become very light, and the artifact bands, which indicate non-specific short molecular DNA, have also increased in intensity. And this is even more prevalent if we look at the I'm so sorry. There we go. This is even more obvious if we look at the sepia that we produced in the labs ourselves. So the bands were already kind of blurry to begin with, and the aging further made them even blurry and harder to identify, to the point where this sample, for example, PC18, had to be considered a negative result. So there is an effect of artificial aging on the probability that we were able to positively identify the squid ink in our samples. So in addition to the swab samples, we also tested the effects of excess sepia in our sample solutions and how it would affect the PCR reaction. Since eumelanin is one of those known PCR inhibitors, we suspected that high concentrations of eumelanin might adversely affect the reaction. And for this, we made one base solution of crema sepia in water of about 1,000 parts per million, and then subsequent dissolutions of the same base solution to see at what dissolution we were able to positively identify sepia. And it might be a bit hard to see, but here these last two samples were the only ones that were positive for the squid DNA in our samples. So I wanted to highlight these results for two reasons. First of all, that the positive identification of DNA in our samples isn't so much a question of how much DNA is contained in the sample to begin with, but also how much PCR inhibitors we have in our samples. So when we try to apply this to cultural heritage objects, we have to keep in mind how to handle inhibitors. And second of all, I want to highlight this because it shows the extreme sensitivity of the method. So these two successful samples down here have a concentration of 4 to 1 parts per million of sepia powder in the solution, and they were still able to yield clearly identifiable bands. We also did some experiments to investigate whether cleaning, like dry or aqueous cleaning, would adversely affect the identification rate of the PCR. And there was a clear effect of dry cleaning and aqueous cleaning, making the samples very, very hard to identify. So for this reason, we are currently investigating alternative sampling methods that would allow us to extract DNA not only from the very surface of the fiber, but from further within the fiber structure. What we're currently looking at is a sampling method that would include placing small discs of agarose gel on top of the ink and then extracting the DNA from those agarose gel discs. However, this part of the research is still ongoing, and the results are as of yet outstanding. So I want to end with a short summary. So I think the most important result that we were able to gain is that swab sampling on its own was able to give us successful identification of squid DNA in our samples. There is an effect of thermal aging as well as of cleaning treatments on the success rate of the analysis. However, the fact that only swab sampling already gave us positive results is very promising, and we hope that we can be able to progress this research in the future. I want to point out the high sensitivity of the method. So with this high sensitivity, we might be able to conduct sampling not from the objects themselves, but from the storage environment of the objects. And yes, there is a possibility of false negative results, so it always should be something that we combine with other analysis methods, and it does require contact sampling, so it is not ideal. Having a spectroscopic methodology for identification would be preferable. Yes, so at this point I want to thank the Japanese Ministry of Education, Culture, Sports, Science and Technology for providing the generous funding that allows me to do my research, as well as thanking Professor Tsukada and Professor Oshikane, who, even though they were not able to attend today's conference, have given me a lot of support and guidance, and I hope that they will continue to do so on this ongoing adventure. I also want to thank everybody in the audience for your kind attention, and I would be happy to hear from anybody working with relevant collections who might be interested in exploring this method with us. Thank you very much.